Good afternoon, everyone, and welcome to the International House. I'm Professor Ksenia Chmutin. I'm the director of Lab for IS. Wonderful to see so many of you here in the room today. Welcome to those joining us online. As always, we run a hybrid seminar today, so the presentation will be recorded and posted on our YouTube channel and as a podcast, but the Q&A session will not be recorded. And I was promised there will be time for Q&A. So welcome to our fellow today, Greg. Wonderful to have you here at Lab. Jakob, please introduce Greg. We look forward to your presentation.
Sure. It's my pleasure to introduce Gregory Pearcey, who's currently an assistant professor at Memorial University in Canada. And Greg and I have connected to work primarily on shifting, on decoding the spinal motor neurons and translating it from isometric actions to movement, which I'm sure Greg will talk more about that today in his talk. So,
all right. Thank you very much to both the IAS and to Jakob for the invitation to come here. It's been fantastic right from even when I left St. John's. Everything's been smooth. I haven't really had to think, which is awesome because I often struggle with thinking. I'm going to talk a little bit about some of the work that I've done both through my postdoctoral fellowships and now what we've been doing in the lab recently at Memorial University in St. John's, Newfoundland. And so my lab is the NI4M, and we're focused on trying to understand the neural control of human movement, predominantly through methods of both surface and intramuscular EMG. So the plan for today is to give you a brief introduction about me and my work, talk a little bit about diversity of firing patterns, motor neuron firing patterns across different muscles of the human body, look at the sex and sex hormone related differences in firing patterns, some firing patterns in spinal cord injury, how we can improve those firing patterns in spinal cord injury, some little bits on sensory feedback, and then why we want to move to dynamic motor output, as Jakob's already mentioned. So my research program, I kind of like to think about it in this way. So when I was first applying for my independent jobs, I was living in Chicago, and Michael Jordan's an iconic member of the Chicago community. And so I found this picture. Obviously, it's an iconic picture. A lot of people know this picture if you follow basketball or basketball culture at all. It's Michael Jordan's wingspan. He's got a ball in one hand and an open hand in the other. So what I like to use as a metaphor here is that the open hand is limited performance. He's not holding a ball out there. In between the shoulders is average performance, and then where he's holding the ball is elite performance. And what we try to do in the lab, try that again, usually works well, is we want to take people who've slipped to that limited performance and move them towards that elite performance. And so if they slip too far towards the open hand, sometimes you just want to slide them back toward the shoulder. You also might want to do some discovery science in between the shoulders. How do we improve people's lives who don't have any injuries? And then how do we get those elite athletes out toward that ball so that they can win the match that they're competing in, or whatever the case may be? And so that's kind of the overarching big picture idea of my lab. And how it all got started as I was growing up in this area. This is West Coast Newfoundland. There's about 20,000 people in, you can drive probably 7 hours in any direction, and there's probably 20 to 40,000 people. So it's quite desolate. Not a lot going on, but it's pretty beautiful. And so we can do lots of kayaking, hiking, and all this kind of stuff. When I was real young, I had the unfortunate event in my life where my mother passed away. And so I was 13 years old, and I was trying to figure myself out, and I started going into the gym. So here's a newspaper clipping. I was getting into some powerlifting. And so what happened was I was understanding that I was getting stronger without growing any muscle. And I couldn't really rack my head around it. I was like, why am I, young age there, 15 years old or something. Why am I getting stronger, but my muscles aren't getting bigger? And it was kind of the kind of starter of why I got interested in the neural control of movement. And so I went on, I did a kinesiology degree, became a personal trainer, did all that kind of jazz. But it wasn't until the new dean of our department gave a presentation that I really got hooked on neuroscience. And so she showed a video of these really nice actions. There it is, of soccer players. And well, I call them soccer, you're probably calling them football players. But just watching them and how they move, how they balance, how they can move the ball, but then also move their body in ways to get around defenders, score goals. And it's just incredible, some of these things that football players can do, but other sports as well. Humans, we're just designed for movement. We move so well. There's a lot of smoothness in our movements, and it's just completely astonishing what we're capable of doing. If you then compare us to robots and the very best robotic football players in the world, the story gets a little bit different. So it is coming now. Let's see.
Give it a click or?
Give it another click. There we go. Okay, so these are the top robotic football players in the world. It's 2016 on the sign there, but I think it goes between 2016 and 2019 is when these videos were taken and they were compiled together. These are the best robotic football players in the world. It's the world championships. And you can see they don't quite move as well as humans do. And so what really got me hooked on neuroscience and motor control, neural control of movement, is why can humans do that so well, but we can't engineer a robot to play football in that same way? And so I did a neuroscience PhD, and I started to read a lot. And one of the influential papers in the field at the time was this review by Steve Scott. And he basically breaks down the neural control and movement to a very simplistic diagram. And throughout my PhD and now some of the work I'm doing, I've become particularly interested in this region down on the bottom where you have the interactions between the muscles and the spinal cord, and maybe some of the brainstem parts as well, which is, you probably can't see it here. Can I use the mouse? This region here is the brainstem for anyone not familiar. And so it's these lower nervous system interactions and the interactions with the muscle that I became really interested in. Apologies, the clicker is sometimes not overly responsive. There we go. Okay. So how do we actually produce forces? Some of you are probably familiar, but there's what's called spikes. These are action potentials. They're sent across neurons in the nervous system. They can come from descending areas. So this could be your motor cortex. It could be the brainstem, cerebellum, and they converge onto what we call the final common path, or spinal motor neurons. And so these are a group of cells located in the spinal cord, and they send the final communication out to the muscles where it tells the muscle, you're supposed to do something now. And so when it sends an action potential, the twitch from a muscle is a 1-to-1 ratio with those spikes. And so by measuring activity from the muscle, we can actually learn what the spinal motor neurons are trying to do. I'm just going to use this for now. And so there's all kinds of spikes in here, and it can give us lots of information about the nervous system, and I'm particularly interested in these spikes. And so if we want to modulate forces, we can do that in two ways. You can have the recruitment of new motor neurons, or you can have something called rate coding. And so the diagram here, I'm just going to switch this to the laser pointer. The diagram here shows that when you're trying to increase the force, which is this black line, you recruit. So this is activate or start to use a motor unit at a given force. And then as you try to increase that force, that one starts to fire spikes faster. This is what's called rate coding. And then eventually you'll bring in another motor unit to try to create more force. It's usually a larger one. They're usually recorded in order of size. And so it starts firing action potentials, and they both start to rate code. So that's the basis of all force control. You start to recruit motor neurons, and then you start to fire them faster. And then when they can't generate a force, you recruit even more. Oh, no, it doesn't do that. Okay. So these motor neurons are then the final common path for all movement, and they integrate all the inputs that come from other places in the nervous system, and they eventually create force output through that pathway I've shown before. And so it was initially thought that these motor neurons were highly linear in terms of, okay, you receive a certain amount of output or input, and you need to give that same amount of output. But it's since been realized that that's not the case. And the reason why is that we have something that's called neuromodulators in the nervous system. They predominantly come from the brainstem nuclei, and they can actually cause like a gain control of those inputs. And we can think of that as a volume controller, where when the volume controller is really high, a small input could give a large output. When the volume controller is really low, a large input could give only a small output. And so this has been a focus of mine from my postdoctoral work, and now even so much now. And the reason why is because all these motor commands that I've mentioned previously can be broken down into three components. We have excitation, that's your go command. You have inhibition, that's your slow command. And then you have the neuromodulation, and that's your volume controller. When that volume controller is really low and we inject current into a motor neuron, this is an in vivo prep now if you have an electrode in a motor neuron, what happens is you get a very linear input output. And so this is just showing you that that volume controller actually works. If we turn that volume controller up, so neuromodulation is high, what happens is we get a highly nonlinear behavior. And so what happens is this is just the firing rate of a motor neuron. The black is an injected current. And when you have a linear input with high monoamines, you get this acceleration of the firing rate of a neuron, an attenuation region, and a long hysteresis. So if we want to measure that stuff in humans, we need to access the motor neurons. We don't want to stick needles, like I said, into a motor neuron that's in your spinal cord. It's highly unethical and probably quite painful. So we use something called surface EMG for the most part. Many of the labs here in your university also use surface EMG. And so it's a very crude estimate of motor neuron activity. You can get amplitude and spectral analysis, but it's then influenced by a number of factors, both neural and muscular. And then there's a lot of limitations and misinterpretations that can be made if you just look at the interference signal from this EMG. So single channel EMG can be quite difficult to use if you really want to look at the neural components of movement. And so what both Jakob and I and many others around the world now are using is we're trying to take that surface EMG signal several channels, and we want to go back to the motor neuron output. So the typical way that people did that is we took a needle, we stuck it into the muscle, and we looked at the spikes by measuring, basically taking a microphone and putting it right next to someone's face and listening to exactly what those motor units are telling us. A non-invasive solution has been developed by Alish Kolobar and colleagues over the years, where you take many channels of surface EMG and you treat it like a cocktail party problem. So if everyone in here was speaking at the same time, it'd be very difficult to discern a single voice. However, if we put in many microphones and we used an algorithm to look at both how far away they are from each microphone and how much time it took for that sound to get to the microphone, we could actually triangulate individual voices in the room. And so that's the basis of a lot of the analysis I'm going to talk about today. What we get then is someone can produce a force output, which is shown in this black line, and then the individual colors are just motor unit firing behaviors, which represent motor neuron firing behaviors of these coming from the spinal motor neurons. And so what you get is many different motor neurons. You can see that they're recruited, so that recruitment pattern I told you to develop force, and then they reduce their spacing in between, which means that they are firing at higher rates. So they have recruitment and rate coding properties. Sorry about this. This is a Mac image, and apparently PC doesn't like Mac sometimes. So this is the image that I showed you earlier. You can't really read it, but it's essentially that injected current, and you have a cat motor neuron firing. So you have various properties. If you compare that to an individual motor neuron firing or a motor unit firing pattern in humans, what you see is you get the steep acceleration, attenuation, and long hysteresis. And those properties tell us about how we control motor neurons in humans. What we can then do, again, you can't read this, I do apologize, is use multiple motor unit firing patterns to estimate biophysical properties. And so the one we're really interested in, which I'll predominantly focus on today, is something called delta F, which is a paired motor unit analysis technique that allows us to estimate that neuromodulation component of that motor command. And so the bigger the delta F, more neuromodulation, smaller delta F, less neuromodulation. There's some intricacies in there that I'm kind of glazing over, but we can get into those in the Q&A if needed. And so what we've been interested in at the end of my postdoc was trying to examine the motor neuron firing patterns across different limb muscles. So for example, you have these proximal muscles that are really important for gross motor control. So that could be holding your arm out while you do some tasks. So if you're putting a key into a lock, they'd probably just hold your arm in place while you do the fine motor control with the tips of your fingers. Likewise, if I'm drinking from a glass of water, I have the gross motor control in the shoulder, and the more fine motor control prevents me from soaking my shirt. So that's what we want to get into here. And so we basically put someone in a chair, we measure from, I think it's 7 different muscles, isometrically, and then we use that technique of blind source separation to look at the individual motor unit firing patterns across these different muscles. So we come back to this, I'll just simplify it down into 2 muscles, the shoulder, so the deltoid muscles, and the FDI, which is a muscle that just moves the finger. And you see that the gross motor control muscles, their individual firing patterns look like this. So each of these lines is an individual motor unit firing pattern. And the task is simple. It's just a linear increase to 20% of maximum, a linear decrease back down, and about 10 seconds up, 10 seconds down. We do the exact same task with the finger muscles, and the firing patterns look vastly different. So one thing I'd like to point out is that with the shoulder muscles, they recruit, but they don't rate code a whole lot. They have initial acceleration in their firing rate, but then they actually have negative rate modulation as force is going up. The finger muscles are more of the classic example, where you recruit them and they increase their discharge rate up to maximum. And so if we just quantify those properties that I spoke about a few moments ago, the delta F values, what you start to see is that there's a linear decrease from the shoulder muscles out to the finger muscles. So what we're trying to get at here is probably there's more brainstem control of the proximal muscles, which allows you to not really think about them during tasks like drinking from a glass of water, whereas your fine motor control relies more on the cortex rather than that gain control to activate them, which allows you to do these, very specific manipulations. They have more direct control from the brain. So when we were doing that study and a couple studies prior to that, we started to notice that all the literature was from males. And so a PhD student I was working with at the time wanted to get into studying females. And so this graph basically shows you that out of 59 high-density surface EMG studies, only 6 of those had equal to or more females for studying motor units. And so this is from 2022. It's getting a little bit better since, but still not a lot of data on females. So that you probably know this literature better than me, some of you, but in terms of performance, there's a huge advantage in males. They have longer limbs, more skeletal muscle mass, lower percent body fat, and higher aerobic power. And so they have a huge competitive advantage in sport, which has been kind of understudied in recent decades, but it is accelerating, which is quite nice. But so there is precedence to study motor units in females, obviously, and so that's what Sophie Jens, who's a PhD student working with me and CJ Heckman, has been working on for the past little while. And so she's looked at the effects of biological sex on motor neurons, and she's found that typically motor neurons in females fire at slightly higher rates. They have much larger estimates of PIC, so more of that neuromodulation command. But they're very difficult to identify motor units from. For whatever reason, we haven't gotten to the bottom of it yet. Female motor units are hard to identify. And so she identified this stuff in 2023, and we really wanted to look at what mechanism might be contributing to these differences. And the first thing that jumped out at us was sex hormones. And so she said, she looked in the literature, she noticed that circulating sex hormones, estradiol, progesterone, have these strong interactions with norepinephrine and serotonin. And norepinephrine and serotonin are actually the predominant neuromodulators that I mentioned that are released from the brainstem. And so what she did is she initiated a multi-site trial. It's now called MUSH, a recent name. And it's basically trying to quantify female physiology across the menstrual cycle at the motor unit level. And so anyone who's not familiar, this is the typical hormone profile of females. So you have the menses phase, where both estradiol and progesterone are low. Then during the follicular phase, you get a increase in estradiol until the ovulation period, where estradiol drops rapidly and progesterone starts to rise until you get out here, where you have high progesterone and high estradiol. And so we get these really unique hormone profiles by measuring people across this menstrual cycle. If we measure them during menses, they have very low estradiol and progesterone. Measure them at the late follicular phase, just before ovulation or close to ovulation, we have very high estradiol but low progesterone. And then if we do in the luteal phase, we get high progesterone and elevated levels of estradiol. And so what they've done now is they've recruited about 50 females, and they've tracked their hormone cycles for 2 months in Chicago, and then 1 month with the groups here in the UK. And then each session they take blood draws and use mass spec to quantify those hormone levels. The protocol was simple. They again do these very simple isometric contractions and then identify motor units at each time point. And so what we found is that the hormone profiles in females are quite variable, and this is well documented in the literature. And so what we tried to do on our first passive analysis is to eliminate people who do not have typical hormone profiles, and that's just elevated estradiol, just like I showed you in that graph. So these red ones were ones that weren't typical within the what would be considered normal levels. So we eliminated those, and we were left with 28 females across the menstrual cycle that we wanted to do a first pass analysis on. And so what we found is that this value, again, I'm just focusing on delta F. We have many other measures, but I'm just focusing on the delta F for today. We found that when estradiol and progesterone are low, we have values that look something like this. There is not much of an increase when people go across intensities from 30 to 50, but then they had this large increase when they go up to higher force levels. When estradiol levels are the same, elevated, there's not much change at this 30% level, but there start to be more excitability or higher values at the 50%, and then they increase to 70, but it's no different than the early follicular phase. Where the big differences occur is this mid-luteal phase. So this is where your progesterone and your estradiol levels are high, and what we see is this overall increase in excitability at all contraction intensities, showing that there's probably some sort of interaction between progesterone and those neuromodulator commands that are being released from the brainstem level that is then able to facilitate the output of the nervous system. And this is literally was sent to me last night, so very fresh off the press. They're still working through some of this stuff, but starting to see some differences across the menstrual cycle. So it looks like progesterone might be where we see the biggest differences in the excitability of motor neurons. Okay, so we're going to get away from females and move into neurological impairment for a few minutes now. So why would we care about SCI, spinal cord injury? Obviously, it affects a lot of people, and those who do affect are affected quite drastically. And so, like I said earlier, all human motor commands are comprised of 3 components. You have that go, slow, and volume control. And when you have a spinal cord injury, you have multiple pathways that go down to the spinal motor neurons, and then you have motor neurons that innervate muscle. After spinal cord injury, the corticospinal and reticulospinal tracts are typically reduced in their ability to activate spinal motor neurons, so those commands get smaller, and our volume control also gets smaller. And so these changes can both modify the number of motor neurons that are able to be activated with voluntary effort, but also it changes their recruitment thresholds, it changes their firing rates, but it hasn't been very well explored. So PhD student Alex Benedetto, again working with Monica Perez, CJ Heckman, and myself, has been exploring this in spinal cord injured patients, particularly the motor unit firing behaviors. And so here's just some demographics. He's got a non-injured control group, and he's got a spinal cord injured group. Ages are very similar. Obviously, there's more time since injury of the SCI group because they don't have injuries. But what you'll notice is that overall, some of these folks are quite a bit weaker. These are just males and females. So this is just their maximal voluntary contraction of elbow flexion and elbow extension. And there's a lot of heterogeneity of the spinal cord injured patients compared to the non-injured controls. The number of motor units analyzed, we're going to skip over the holds. I'm not even going to talk about that today. But for the contractions where he examined delta F scores, which I'm going to talk about today, you can see that even with similar amounts of participants, he was able to identify similar amounts of motor units in his injured group compared to his non-injured group. There is a reason for that. It doesn't necessarily mean that they had more motor units active, but with spinal cord injury, sometimes the signals are more sparse, meaning that there's not as many active, and that can actually help with our identification methods. So again, very similar protocols to other things I've talked about so far. This is the setup that people were in, and he had people either do flexion or extension, and then we do our typical analysis. If we look at the ability to change the discharge rate, so that's that firing rate modulation that I talked about earlier, or rate coding. People with spinal cord injury have drastically reduced ability to modulate their firing rates. So they don't have that synaptic input to be able to really modify that force output. And that really shows in their ability to cause force output. But the other thing that's quite interesting is that we were able to stratify, these are individual motor unit values here, and each basically vertical column is motor units identified from a different person. The x-axis is their maximal force output from, on the left, biceps brachii, and then the right, triceps brachii. So this is essentially flexion, extension. And here's the delta F values. And what we noticed is that there was a strong positive relationship using a generalized linear model of your ability to have force output and the neuromodulation that's coming down to those motor unit pools. And so we thought that this was quite interesting because it starts to give us a biomarker of recovery of function after neurological injury. And so now what I wanted to do during my postdoc is how can we use some of this information to actually improve function? And so searching for a method to do that, we came up with a therapeutic intervention that's been used for many years in respiratory world called acute intermittent hypoxia. And so how this works is people breathe short periods of 9% inspired oxygen, and then they interleave that with 21%. So that's room air oxygen. And they can do this either in a control condition or sham condition where they just breathe. We just switch them back and forth between different tubes of room air, or we give them this low oxygen air. We can do strength tests before, strength tests after, and then we just essentially use those same methods I've been talking about all day. How do we look at that motor output from these individuals? And so how it essentially works is this is the inspired oxygen is in the red here. This is a lag in the oxygen saturation that happens usually after you have these periods of oxygen. It's almost like doing sprints, except you're not actually moving your body. You sprint, your SpO2 drops down, you rest again, it comes back to baseline levels. You sprint again, it drops, it comes back. And you do that for 15 cycles or something like that. It's like doing 15 sprints. And so what this does is this cyclical reduction in SpO2 actually stimulates the carotid body chemoreceptors, which have direct activation of the brainstem nuclei I've been talking about all day, which then facilitates downstream serotonin signaling pathways, which alters the motor neuron excitability. So what we've shown for a couple different studies now is that when you have people produce elbow flexion and extension force, this is just showing the flexion here. On the top, we have maximal force output in spinal cord injured patients. This is before they receive any acute intermittent hypoxia. This is immediately after, and this is 1 hour after receiving acute intermittent hypoxia. And here's the intensity of muscle activity across that grid. So you can see their intensity of muscle activity after receiving this treatment within an hour is largely facilitated. So we wanted to get down to the mechanisms. Like I said, surface EMG has a lot of limitations, and we might not understand why that's actually happening. So what we wanted to know was, are they better able to change the discharge rate in their motor units during maximal efforts? And so in collaboration with Aleš Holobar of Slovenia, who's been kind of leading the charge in these identification methods, we actually looked at individual motor unit firing behaviors during the maximal contractions. We did submaximals as well, but we didn't find anything interesting. So in our paper, we only actually report on the maximal ones. And so here's prior in an individual motor unit firing behavior. Here's prior to any acute intermittent hypoxia. Here's the maximal force that people can produce, and here's the discharge rate. And then we follow that same unit across that 1 hour period, look at the discharge rates again. Their forces are facilitated, and so are their discharge patterns. So we're able to use that therapeutic intervention to facilitate the motor neuron output. And so you might say, oh, that's really interesting. There's not a lot of spinal cord injuries in the world. You might help some people, but what about a more common type of neurological impairment? And so we found that it's helpful for SCI, but what about, like I said, something else? So what we did is we took in a group of people who were living with chronic stroke. So they've had a stroke at least 6 months prior. And we basically did a titration protocol because when you're dealing with an injury that was caused by ischemia, you don't want to cause more ischemia, which could further injure them. So what we did is we took them in, we started them with breathing room air, brought them back 2 days later. We had them breathe 17% oxygen, brought them back again, 13% oxygen, and brought them back again, 9% oxygen. And we had them hooked up on it was in a hospital, Schiller Ability Lab. We were measuring all kinds of vitals. There was a nurse nearby and a neurologist nearby. And so what we then did was looked at function of these people. And so this was just recently published in Stroke. So by using this same protocol where we have 30 seconds of low oxygen air, there was no changes in physician-assessed signs and symptoms of neurological impairment. They didn't have any negative changes when we performed these titrated bouts of breathing. However, they did see vast improvements in the grip strength and elbow flexion force. That's the only two we measured. In their more affected side. Their less affected side, it didn't see any changes. But the more affected side, there was big changes. I don't have the percentages listed here, but you can find those in the paper. So it was really quite interesting. So acute intermittent hypoxia, it's very much understudied, but does have a lot of potential. Okay, so another topic, kind of jumping around a little bit here, but it'll come together at the end. All of those pathways I've been talking about so far is mostly brainstem input, it's descending input, and then it's force output. But what we're kind of neglecting in a lot of these studies is the role that sensory feedback also plays. So these are typically afferents, so sensory neurons that sense your speed of movement, your skin interactions with the world, the amount of force you're outputting. We kind of neglect all of that in the studies that I've already shown you so far. And so one thing you can do that's really cool if you put a blood pressure cuff on someone and you inflate it and just leave it on there for a long period of time is that you can actually get a point where these are your motor action potentials and these are your sensory action potentials. You actually get a period of time after about 30 minutes, it's quite variable between participants, where you get motor action potentials that can pass that cuff, but sensory action potentials can't. So you get a really neat area of time or period of time where people have full motor function and no sensory function. And so this is kind of one of the first projects that Jakob and I have been starting to work on recently. And what we can show is that when we inflate that cuff for a long period of time, we can use an H reflex, which is a spinal reflex, to give us an idea whether there's sensory transmission. And then over that period of time, you get no sensory transmission, but the motor waves can still pass. That's the M wave. So a little bit simplified, but you get a period where there's motor function and no sensory function. What happens during this is that the maximum discharge rate of people and their ability to produce force when you take away sensory feedback is vastly reduced. So here's the black line is their maximal force output, and these are just different motor units which have been identified during maximal behavior, and they're firing at a kind of an average rate of this dotted line. When you take away that sensory feedback, the force is reduced by almost half on average across people, and those discharge rates are vastly reduced as well. So you can't maintain discharge rates when you don't have sensory feedback. You also can't control forces very well. So here's an example prior to occlusion. Here's different motor units firing, and here's someone's force. Here's when they have no sensory feedback, and you can see that these force changes are really, really big, and they have a lot of intermittent firing. Again, showing that you can't maintain motor neuron discharge when you don't have sensory feedback. As soon as you take that cuff off, you're almost back to baseline in terms of your force control and your ability of those motor neurons to maintain their discharge rates. So that's taking away sensory feedback. What if we add more sensory feedback that's irrelevant to the task you're trying to produce? So this is work by an undergraduate student in my lab last year who's quite impressive. And she designed an experiment where basically people perform a task you ask them how hard they just produced force, you take away feedback and tell them to produce that same force again. And what she did is she then vibrated various tendons, either the Achilles tendon or the TA tendon, so the antagonist or the agonist tendon. And so what happens when you vibrate the Achilles tendon, so this is the antagonist tendon, you cause a spinal reflex that is very much inhibitory to the muscle trying to produce force. If you vibrate the same side of the muscle, it's technically excitatory, so you can cause an excitatory reflex, but almost more importantly is that you cause a disruption in your ability to sense what's going on. And so what she did then is she had a control condition, so people produce force. They asked how hard they produce force. People would say, 3 out of 10, and then say, okay, do 3 out of 10 again. And they were pretty good. This example, they were really good. We then vibrated the antagonist tendon in the first contraction and then asked them to produce that same force again. Turns out they overpredict the force. If you do the opposite, so you don't vibrate them in the first contraction, you vibrate in the second contraction, they tend to underpredict. And this was then repeated with the tendon vibration. Turns out that the agonist tendon vibration, turns out that doesn't do a whole lot. You slightly overpredict force. So that's in the yellow and the red here are the two vibration of the agonist tendon, and then the blue is the control condition. But the gray, which is the one where I showed they overpredict, and then the green is the one where I showed they underpredict. We're able to access spinal circuits to really get at the perception of force. And so what we started to think about then is, well, we're missing a lot of sensory feedback in all the studies that we ever do, because in real world motions, people are moving. And when you move, you had a lot more sensory feedback than when you do when you're sitting in a chair, your muscle's not changing its length, and all you're getting is the interaction with your skin, with the device you're pulling or pushing against, and then the forces that are sensed within that muscle. And so that really gets into why Jakob and I got in touch with each other. And so in my lab, we're starting to do these very slow dynamic contractions where people go in a lengthening contraction. So this could be going through plantar flexion when you're activating the TA, and we get these really large estimates of that neuromodulatory command. Whereas then when we do a shortening contraction, so concentric contraction, we get these really small estimates of neuromodulatory commands. And we think that there's large interactions of the sensory feedback that are contributing here. And so this is very preliminary work, and I don't have a lot of insights on it, but I'm happy to chat about it if anyone's interested. We're now moving, again, some of the stuff that Jakob and I have been talking about recently is moving from an isometric contraction to walking conditions, and trying to really match the intensity of output between isometric and walking, and then looking at the firing patterns that occur when we do that. And so we've been making some really neat progress with this. So here we have 3 TA motor units identified with their individual signatures, and then 5 Soleus motor units. And what we've been able to do now is using motion capture and these innovative high density surface EMG that are completely wireless, is that we get motor neuron firing behavior during walking with the kinematics that go along with it. So something that was really difficult to do for many years, we're now getting really close to being able to do completely dynamic behaviors and really start to study things like walking after neurological impairment, where there is central pattern generator contributions and sensory feedback contributions. And our ongoing work now is to pair high-density surface EMG with intramuscular arrays. And so here's a student of mine, Ben Nazaroff, and another one, Maddie Hewlin, and then a physician who's just happens to be my best buddy and best man at my wedding, who wanted to get back into some research. So we're doing some ultrasound guided insertions of these arrays into the muscle and looking at different compartments of the muscle. And so this is very much in its infancy and stuff that Jakob and I plan to work on together. And so with the help of Simone Avrillon, we've shown now that rather than just getting, 10 or 20 motor neurons from the surface of the muscle, we can get many more motor neurons or motor units from deep within the muscle. And we're now able to start to look at the behavior of larger populations of motor neurons and how they interact to create these forces, and start to look at some dynamic behaviors. And so just a summary of this recent work, motor neurons are accessible in humans based on their 1-to-1 firing ratio with muscle fibers that they cause to twitch. Firing patterns are diverse across muscles in the human body, and they represent probably a functional role and an anatomical projection to those motor neuron pools. Firing patterns certainly differ between the sexes, and they are influenced by hormones, albeit that work is as much in its infancy. Firing patterns are affected by neurological impairments, as probably would be expected, but we're able to now generate biomarkers to try to look at what might improve function after neurological impairment. And then acute intermittent hypoxia can improve those firing patterns. So therapeutic interventions can be used, and we can actually measure whether something is improving. Firing patterns are also affected by sensory feedback, but we know very little about dynamic motor output, and that is the focus of the collaboration that Jakob and I will have for hopefully many years to come. So lots of people in the lab, thank you very much. This is our annual axe throwing competition, which I had to delay because it's typically this week. The winner here is Emma Mitchell. She's right here. She beat me in the final. And then my dad usually comes out for the axe throwing as well. So we try to have a bit of fun. The other thing is we do have graduate programs now. So if ever anyone is wondering if they want to come to cold Newfoundland in the North Atlantic, give them my contact information. And then this is kind of what it looks like. So here's Signal Hill, a very iconic area of St. John's, and then the Battery. So that's the downtown area of St. John's. It's not the actual city area. It's kind of over off to the left. But this is an iconic picture of St. John's. And then there's really nice trails that go around. So there's my daughter, my friend who I showed you injecting that electrode with the ultrasound image, and then two of our wives and my two of our daughters are on our backs and hike around there. Summertime is really nice for hiking around there. And with that, I will leave it there if there's any questions.